Hi everyone, we're happy to introduce to you today our paper Recovery in Pupillometric Non-Visual Functions Following Chiasmal Decompression in Pituitary Adenoma, recently published in Brain Communications. I am Daniela, PhD student at the National University of Singapore, Department of Ophthalmology, and I am joined by Dr. Jensen Ang, a neurosurgeon at the National Neuroscience Institute, Singapore General Hospital campus. And we are the first co-authors of this paper. For this video, we are using the graphical abstract published with the paper to break down our study. Let's start by introducing or understanding pituitary adenoma and why it is a disease of interest with Dr. Jensen.
Thank you, Dianella. Pituitary adenomas are among the most common brain tumors. They grow just beneath the optic chiasm, the crossing point of optic fibers coming from both eyes. And as they push upwards, the pressure cuts visual signals, leading to vision loss. But the optic chiasm also carries fibers from a specialized retinal cell called iPRGCs, expressing the photopigment melanopsin. These cells drive the pupillary reflex, as they wire directly into the brain circadian clock. One way to treat pituitary adenoma is through endonasal transsphenoidal surgery. This is performed through the nose, and the aim of the operation is to decompress the optic chiasm. On the MRI, we can measure the decrease in the upward displacement of the chiasm post-operatively, as well as the increase in the optic chiasm thickness. But here's the problem. Structure and function don't always recover together. Some patients regain their visual fields, but others don't, even when the MRI looks perfectly normal afterwards. And right now, we have no rapid, objective bedside tool that can track this functional recovery, visually or non-visually in real time.
For this study, we recruited 27 patients with pituitary adenoma causing chiasmal compression with 41 healthy age-matched controls. Patients were tested twice, before and after surgery, with MRI, visual fields, OCT, and handheld chromatic pupillometry. Our pupillometry protocol takes just 1 minute per eye. The device shines blue light and then red light into the eye, and a built-in infrared camera records the pupil's response. The initial fast constriction that you can see on the graph reflects your cone photoreceptors' contribution to this pupillary response. But what's more interesting is what happens when the light goes off. The post-illumination pupillary response, or the PIPR. That sustained signal is mainly driven by melanopsin. So in only 60 seconds, we're probing both the visual and non-visual photoreceptors simultaneously. But now the question is, did surgery restore these responses? And did they recover equally across both pathways? After surgery, pupillary responses did improve, but not equally. Maximum constriction to both lights got better, but it still stayed below control levels. The cone-mediated responses recovered partially, mainly in patients with visual field recovery, but the PIPR fully normalized and recovered after surgery. And this happened regardless of whether visual field recovery occurred or no.
We also asked whether structural recovery on MRI correlates with functional outcomes, and the answer is yes. Greater chiasmal decompression correlated with better visual field scores, as well as with stronger cone-mediated pupillary responses. The more the anatomy recovers, the more the visual pathways will follow.
So what does this mean in practice? Handheld chromatic pupillometry provides something we've been missing, a simple, 1 minute, objective and non-invasive way to track both visual and non-visual recovery. There's much more information in the paper published in Brain Communications, so please check it out. And do feel free to contact our corresponding author, Dr. Raymond Najjar, if you have any questions.